served and are given numbers like P8, P7, P3, and so on. So you get the idea that this is a very highly kind of conserved structure, and that mutations that would be in these consensus sequences might very much affect the function, the self-splicing of the intron. Now, this is discussed in your textbook, but kind of not very well. In fact, it's kind of so misleading that I like to go through and talk about kind of the spots in the textbook where it's a little bit tricky, a little bit iffy, okay? And so this is a figure out of the textbook, and it really shows what I've just shown you with a little extra part to it, which also is something, reason to kind of show this. So here is exon 1 and exon 2, and the intron that we just talked about, that black piece that's removed, is 408 nucleotides, okay? And here is the A nucleotide, G nucleotide that I talked about in the previous transparency. And the first transesterification step here uses the G nucleotide, which is talked about here. But what I don't like about this in the textbook and the way this is described is that this arrow says cleavage at 5' splice junction and G addition. And that's kind of the end result, okay? And here it says cleavage at the 3' splice junction, ligation of exons, but you have to realize that this is happening as a transesterification step, okay? The newer text talks about this a little bit better than the old textbook does. And so I just don't want you to be tricked by this, because one of the things that really tricks people a lot about this figure in the textbook is that so this is the first transesterification step, and then the OH here attacks here, and you get the exons joined, and here is the G nucleotide, and here's the excised intron, okay? And that's, from what I just showed you, that's where the things end. But now it turns out that there is in this example, there is a third transesterification step, a triple transesterification, okay? Now this really doesn't have anything to do necessarily, it just kind of happens by chance because it doesn't have anything to do really with the excision of the intron here. But what happens is that this structure that is in the intron is still formed, and it still has its ribozyme characteristics. And so it can do a third transesterification, and that's what's happening here, but it's not illustrated very well. And so this G nucleotide is attacking a phosphodiester bond here, and so there ought to be an arrow with this G. And then the G becomes linked to the side, so you get a circularization of this piece right here, and this little piece gets cut off and has an OH group at the end here. Now that's not shown very well, and what really makes me crazy is that they make this look like a lariat structure. And a lariat structure happens in spliceosomal introns. It does not happen in group 1 intron, and don't get the idea that this figure is showing you that. It's just not a figure that's showing you that you can get circularization here, and this is a third transesterification that makes a new bond here to circularize this, leaves an OH here, and that there is an OH, and the OH is at this 3' end. This G here is this G here, and it's using this OH to attack a phosphodiester bond that's right there. Okay? So A, it's not cleavage and ligation. We'll talk about that kind of mechanism later. It's a triple transesterification where that last step is not involved in excising the intron, but it is involved in circularization of the intron, and that happened and mystified them at first when they were trying to figure out the mechanism. Okay. And I might mention just that this turns out to be, the circularization, it turned out to be kind of nice for molecular biologists, because this circular part still retains the three-dimensional structure of the ribozyme and actually can act as a transacting enzyme or ribozyme, okay? Because it is able, it will able to like bind substrate just like a real protein enzyme, that was very, very useful in kind of trying to study the mechanism of ribozymes. Okay, group 2 introns. Okay, well, the mechanism for group 2 introns looks like this. Okay, I'm not even sure if I have this in your notes as a separate piece, because those of you might notice that this is exactly the same type of mechanism we talked about for spliceosomal introns. So this is easy. It's the same as spliceosomal introns. And so let me just review this real quick. Is group 2 introns have a 2' OH here on the A, and that then attacks this phosphodiester bond to join the G to the A in a 5', 2' linkage right here to make this lariat. So group 2 and spliceosomal introns make lariats. Group 1 do not make lariats. Okay? And so this is the second transesterification step that we talked about, joins the exons and excises the lariat with a 3' OH. All right. So, what makes this different from a spliceozyme? A spliceosomal intron. What makes it different? Well, what makes it different is the fact that this one is also self-splicing. Okay, and so again, you've got important stuff in the intron that is necessary for the self-splicing, the self-excision of its own intron. So this intron exists for the primary purpose of removing itself from an RNA. And if you think about that, that's kind of, that doesn't kind of make evolutionary sense. But in fact, in a way it does, because people have at least proposed that this intron is what's called selfish DNA or selfish RNA, that it's perpetuating itself by getting replicated along with the DNA of an organism, and that it has a mobility. It can kind of jump to other sites, and if it goes to other sites, then it could go into the middle of a gene and disrupt it. So it had to evolve a mechanism, or when it evolved, a mechanism so that it is removed without disrupting the mRNA or the ribosomal RNA of that gene, then it could be perpetuated. And so we'll talk about the fact that there is mobility. Okay? So actually, that's not really a weather alert. Every time I mention a test question, a beep goes off. Okay, so. Okay, so the structure that's necessary for group 2 introns in the intron is also understood and is shown here. And you can see that it doesn't really look anything like a group 1 intron. And it has a very, very kind of, again, recognizable structure. This, you can kind of draw it in a circle with the blue here. I have the blue exons kind of close together. And the phosphodiester bonds that are attacked in each transesterification step are shown with red arrows. And so when you organize it like this, you end up with these kind of double-stranded RNA regions, these duplexes, coming out of the central region. And these are sometimes called radial spokes. And there are 6 radial spokes, kind of like the spokes in the wheel, and they're going out from the center. And you can see that some of these regions here are quite large, and so the loops in these stem loop structures can be quite large, and they are going to be important later. I'll just mention the fact that they provide space where you can actually have protein coding genes in them. Okay, and so we'll talk about what those proteins might be doing a little bit later, but just kind of remember that. And then you have 1, 2, 3, 4, 5, 6 of these. 5 and 6 are really quite small and conserved. And I want to really focus in on 6, because 6 has this A nucleotide here. And this A nucleotide is the bulged, again, it's a bulged A. And just like in a spliceosomal intron, this A's 2' OH will attack this phosphodiester bond here and connect this G to the A through a 2', 5' phosphodiester bond, 2', 5'. Okay? And then the second, so that leaves on the 3' end of the 5' exon, on this C, there's now an OH, because it's no longer connected to the G here, and that OH attacks right across here and connects the C to the A to link the exons, and that leaves an OH on this U right here, and so these become linked, and this whole part of this is excised. Okay, so this is group 2, it's self-splicing, and it has the same double transesterification mechanism as a spliceosomal intron, except a spliceosomal intron doesn't carry around its own splicing apparatus, whereas this intron does. Now you might kind of think that that makes these, the group 2 and the spliceosomal introns, they're really quite similar, and you might think that they're kind of evolutionarily related, and there is evidence for that. And just to kind of show you how group 2-like introns, including spliceosomal introns, are similar, it's showing consensus sequences here. And I just want to mention these consensus sequences really briefly, because there's kind of 2 points I want to make with them. There are 2 points I want to make. One is that they have very similar consensus sequences. And so these top 2 are group 2 introns, one in chloroplast and one in mitochondria, and just show the 5' and 3' junctions. And then compare that with our consensus sequence that we've been using for spliceosomal introns. So the bottom 2 are spliceosomal introns, and that this, kind of a little hard to see, but this is the consensus sequence that I haven't shown you before for the branch site, and it's showing it for the branch site for yeast and for kind of the general nuclear spliceosomal introns, and a branch site for a group 2. Okay, and these you can see are very similar. Now another thing is I'm showing you this for the reason I'm showing you this is that one of the problem sets that I'm going to give you, and I don't think I'm going to assign it today, but probably at the next meeting on Monday, requires you to use the consensus sequences here to solve the problem. Excuse me. And you don't have, I don't want to imply that you have to memorize these in any way. I just want to say that you'll need to use those to solve a problem. And in an exam, if you need these kind of sequences, I'll provide them. Okay, but and what these green numbers here mean is kind of to show that there's just a little bit of space. The spacing is a little different. It's real close. The branch site is real close, 5 to 7 nucleotides to the 3' consensus. And here the branch is pretty close, but not quite as close, about in the order of 22 to 37 nucleotides. So they have a Lariat intermediate, and they have similar consensus sequences. And so there is some, at least, circumstantial evidence that group 2 and spliceosomal introns probably maybe have a common origin. Okay, this is a kind of an interim summary transparency. I think there's nothing new on this transparency, but I want to kind of just go over it with you because it is so good to study this transparency because it has a lot of nice kind of testable information on it. And so it is a comparison of group 1, group 2, and spliceosomal introns. And so here you have the intron, and this is kind of, this green symbolizes that there is a kind of a secondary structure in these two. They're different but important. And here there is this big purple circle that is supposed to be the spliceosome, or to remind you that there's a spliceosome necessary here. Okay, so first of all, these two are self-catalytic and this one isn't. And then it shows the first the first transesterification step in both of these, and in each case it is an attack of an OH and a phosphodiester bond. Here it is a G nucleotide that is bound by the intron in the group 1, and it attacks at this phosphodiester bond, the upstream phosphodiester bond. And here the A is using its 2' OH here to attack this same bond. And here again, this A is attacking, is using its 2' OH to attack this bond. Okay? So all of these do a first transesterification step, leaving an OH on the upstream exon. So that's the same in every case. And here you have the G nucleotide attached to the intron. It's not a lariat, this is linear. This one is formed a lariat because it has a 2', 5' junction, and this one has a lariat with a 2', 5' branch. Okay. And then the last transesterification step is shown here, and it's the same in every one of them. This OH here, here, and here attacks the phosphodiester bond right here, just upstream of the downstream exon, and fuses the exons in a 5'. All three of these are 5', 3' phosphodiester bonds, and excises the intron as a linear here, or as a lariat here. Each one of these have a 3' OH. Okay, but this one is a lariat, and both of these would need debranching enzymes to break them down. This one would not. Okay. First interim summary. Any questions on that? Okay. If you don't get a feel for the fact that I want you to kind of really understand double and triple transesterification steps, let me tell you that I really do. Okay. Yes?
You said that the group 1, the intron is linear, but the picture you showed was circular.
Yeah, and that's a little tricky, but if you, but there's no connection here between the end and here. So this is linear. These two are the lariat forms where there is that 2', 5' branch.
In the picture you showed from the book, you said it's not a lariat.
That's right. So group one is not a lariat, and it's linear. And then, so that's what I'm showing here, okay? So this is, maybe to answer your question, the circularization step, which would be downstream of this, is not shown in this figure, okay? I guess that's what you were asking. Any other questions? Yes?
The lariat from the nuclear mRNA, is it just started outside of the nucleus there? Is that what it's showing, that it just started outside of it, or is it still inside that lariat at the end that you get? For both products?
Say the question again, because this is not the nucleus. Okay, this purple circle is the spliceosome. Okay, and indicates that it's not self-splicing. Okay, although your question is kind of is valid, because in some cases the excision of the group 1 can happen in the nucleus, and it is then it is just turned over and degraded in the nucleus in those cases. Any other questions? Okay. All right. Well, I want to kind of now switch gears from. I'm not going to quite leave splicing yet, but I want to kind of leave the double transesterification type of splicing and talk about another kind of splicing. And before I do that, I want to start talking about tRNA maturation, the processing of tRNAs. We've already talked just briefly about the processing of ribosomal RNAs. We'll talk about that more in just a moment. But I want to talk about tRNA maturation or tRNA processing. And so kind of symbolically here, what you're seeing is that you have a tRNA, and tRNAs are not produced de novo as cloverleafs, but in fact they have an extension on this cloverleaf, a 5' extension and a 3' extension. Okay, this is sometimes called a leader sequence, and